example is the cell will grow on two different sugars, glucose and fructose. And there's a gene that codes for a cell surface transporter for each sugar. Now, if you knock out the glucose transporter gene and there's both sugars in the environment, the cell lives. So we would score that gene as a non-essential gene. But if you do the same experiment with only glucose in the environment, the cell dies. And so that becomes an essential gene. So we found that we can only define organisms if we have two components. We have the genetic code and we have the environment described. Obviously, the simple part of this is defining the genetic code. Because we can only knock these out one at a time, and if you look at the metabolic pathway here, it's relatively simple compared to any of our cells. But if we look at all the genes that could be knocked out one at a time, this would be a non-functional cell. So there's far more here than just the conditional except functional genes. So we decided the only way we could answer these questions would be to make a synthetic chromosome where we could deliberately vary the gene content of this species. This created new questions. Would chemistry even permit us to make these large molecules? And even if we could eventually make them, how do you boot it up? Would it even be active if we start with 4 bottles of chemicals? Well, to make a very long story short, we spent years doing this, but before we ever started these first experiments, we asked a bioethical team at the University of Pennsylvania to answer the question for us, was it okay to try and create life in the laboratory? They were stunned by this, but spent a year and a half on the problem, bringing in all kinds of groups, religious groups, scientists, lay people. And they published a report in Science in 1999 saying we were approaching things on a reasonable basis. It was actually very difficult to synthesize DNA. Our DNA synthesizers have a degenerate process. It's an N-1 situation where the longer you make the piece of DNA, the more errors there are in it. It's not a problem if you're just making PCR primers of 20 to 25 bases. If you're trying to make a 500,000 base pair genome, it's definitely a problem. So my colleagues in this are Ham Smith and Clyde Hutchinson, and they developed some novel methods of error correction. And we decided to start with Phi-X174, a bacterial phage or bacterial virus that kills E. coli. This was the first genome that Fred Sanger, in fact, had sequenced, and Clyde was in his lab when he did that work. So we resequenced the genome to make sure we were starting with accurate digital information. And through this process, it only took about 2 weeks to chemically make the 5,000 base pair piece of DNA. The exciting phase came next. We inserted that piece of DNA into E. coli. E. coli recognized our synthetic DNA as natural DNA and started making proteins. Those proteins self-assembled to form the virus. The virus, being grateful as they are, started killing the E. coli cells that made them. And that's how we can detect them. You get these clear plaques on the plate. So what we synthesized was 100% accurate. And this was a unique situation, if you buy the software analogy, where we view this as the software actually building its own hardware. So we're not starting over back at the origins of life. We're starting after 3.5 billion years of evolution. And we're showing that we can alter the software and go to the next stage. We didn't want to make a small virus. We wanted to make the complete bacterial chromosome to understand these basic components of life. So we thought we would construct it by putting together viral size segments so we could insert genes, leave them out to test what was really essential. We went through a long process. It turns out design is really important. So I was very disappointed in the reports on this lovely work on the woolly mammoth of how that was going to lead to Jurassic Park scenarios. The reporters that I talked to, I told them this was not going to be possible. So if you're hoping to reconstruct biodiversity based on having some inaccurate sequence, don't count on it. The sequence accuracy is really critical. In 1995, when we sequenced the Mycoplasma genome, the state of the art was 1 error in 10,000 base pairs. We resequenced the genome and we found 30 errors, and several of them would have been lethal. So you can only come back from the digital code if that digital code is accurate. And things like the mammoth and some early humans, the data is incredibly incomplete. So we started with new sequence in the computer, and we had to design it so we could make small segments that would overlap to form larger segments that would overlap to form even larger segments. And one of the things we worried about as we're working on this frontier is what are the artifacts that could be lethal to our careers if we thought we had booted up a chromosome, in fact hadn't. It turns out one single molecule of native DNA could have fooled us. And so we devised a unique way to test for this. We decided to watermark the genetic code that we were synthesizing. Now, if you're a molecular biologist, you can view this as fun. You know, we have a 4-letter code, A, C, G, and T, and we all know that triplets of that form our 20 or so different amino acids. That there's a single letter designation for each amino acid. So in the genetic code, we can write words, names, sentences, anything we want. Actually, I got the idea for this originally when we were in the midst of sequencing the human genome, and somebody from Hollywood brought me a script and wanted me to advise on whether they should make this movie. The guy who was sequencing the human genome discovered the book of Genesis was written in the genetic code. And I thought that was kind of a novel idea. And I said, well, what happened to this guy? They said they killed him off right away to make a good plot. I said, I don't like this movie. But we found from during that transposon mutagenesis, we knew places we could insert unique text in the genome to watermark it uniquely. And they're shown in these vertical bars on the top line. Now, many people have reported that they've been disappointed that we didn't write something really profound, like one small step for bacteria, one giant step for mankind or something. Ham Smith, Clyde Hutchinson, and others signed our work, signed our names in the genetic code. But I think the chance of those names occurring naturally in a mycoplasma genome are pretty close to zero. Well, we went ahead and made these smaller pieces, assembled them into larger ones. This looks like a basketball Final Four. But the problem is, once we were even at the 3rd step here, we were in territory that nobody had ever been in before. The largest piece of DNA anybody made was about 30,000 letters. So as soon as we're up in the 72,000 base pair range, it became problematic. Once we went over 100,000 base pairs, these pieces did not like to grow in E. coli anymore. So we started looking for some novel systems, and we'd been studying a number of organisms. Early on, we sequenced the organism of Deinococcus radiodurans, which is this organism that can take about 3 million rads of radiation and not be killed. What happens is its chromosome gets blown apart with a couple hundred double-stranded breaks, like you see in this top panel. But after 12 to 24 hours, it stitches its genome back together exactly as it was before, and it can start replicating again. It turns out this is not such a unique property, although we can't do it. We're learning how to do it in the lab, but our bodies can't do this. A lot of organisms can. The late Francis Crick and others talked about panspermia, and a lot of people dismissed it because obviously no organism could withstand the harshness of outer space. But this is one of probably thousands, if not tens of thousands, that can exist in a vacuum, get really high doses of ionizing radiation, and as long as they get dropped in some water at some stage, they're able to reassemble its genome and start replicating again. So even if life did not evolve on this planet by panspermia, we're certainly making sure it will probably evolve someplace else with the constant spread of organisms into outer space.